Hi everyone, I'm Nathalia. I'm from Federal University of Rio Grande do Sul, Brazil, and I'll talk about my work. Transposable elements abundance correlates with mode of transmission in Microsporidium parasites. So first things first, what are Microsporidians? They are obligatory intracellular parasites and very distinctive organisms. Their cell morphology and biology are so unique that they don't have an obvious relationship to other eukaryotes of the tree of life. So their classification was a challenge. Through phylogenomics, we now know that they are positioned on an ancestral branch within the fungi phylogeny. The smallest eukaryotic genome known belongs to a Microsporidium. Microsporidians went through an extensive genomic and metabolic reduction and lost many gene families. The majority of Microsporidians even lost genes related to TCA cycle, so they have to steal ATP from the host. But even though they are so tiny, there's a huge variation in genome size in Microsporidia clades. Large Microsporidian genomes can harbor a great number of transposable elements, while the smallest genomes are completely devoid of these. We know that when Microsporidians are vertically transmitted, that is, when the mother passes the parasite to the offspring, it produces population bottlenecks, reducing the effectiveness of natural selection in removing mildly deleterious mutations in these parasites like transposable elements. This causes enlarged, less dense genomes with accumulation of these. On the other hand, horizontal transmission, that is, when the host catches the parasite in the environment, is associated to more compact genomes lacking of these. So we decided to characterize the mobilomes of a large number of genomes of Microsporidium. So we screened all Microsporidian genomes available in databases. We wanted to see also if the accumulation of transposable elements is related to mode of transmission. Therefore, we obtained 47 publicly available genomes belonging to Microsporidia. We obtained 47 publicly available genomes, and we searched them for 37 highly conserved proteins through our phylogenomic reconstruction. Next to the species names, we have H, that stands for horizontal transmission, HV, horizontal and vertical transmission, and AGS, horizontal and sexual transmission. Next to it, there's genome size in megabases. And finally, the fraction of each type of TE in the genome of these lineages, categorized as DNA transposons, LTR, retrotransposons, lines, and unknown types. We used softwares Repeat Modular, Repeat Masker, and Mite Tracker. We found out that DNA transposons are the dominant class of TE in Microsporidian genomes, but their abundance varies greatly in lineages, even in the same species, and it's not phylogenetically conserved. To evaluate whether transposable elements accumulation represents ancient or recent events, we used Repeat Landscape, a program inside Repeat Masker package, to calculate Kimura distance between each TE copy in the genomes in relation to the consensus sequences in the Repeat Masker libraries. So here in the graphs show the fraction of the genome occupied by each copy of TE and the Kimura distance to the consensus. So more to the right, near 50, means more divergent from the consensus. Therefore, it's an older insertion. More to the left, near 0, means less divergent from the consensus, so it's a more recent insertion. The lineages with the highest proportion of TEs, like these two, exhibit a recent event of TE amplification. We then decide to test whether there is a positive correlation between genome size and TE fraction in the genomes. So we applied Spearman correlation with R. So as we can see here on the right, there's a positive correlation between TE fraction and genome size, although some strains with large genomes like A. augeri and E. aedes have a fraction of TEs very similar to Small genomes like encephalitoson, funiculi, and here on the left, we can see a strong negative correlation between coding sequences and genome size, which means that non-coding regions are responsible for genome variation in Microsporidium. But why are there differences in T content in Microsporidiums? We reason that these recent events of T amplification in some lineages could result from the loss of genes related to genome defense mechanisms such as dicer and argonaut, members of RNA interference pathway. Therefore, we identified dicer and argonaut in 27 proteomes. But contrary to what we thought, lineages with putative dicer and argonaut have on average a higher proportion of TE and a larger genome size compared to lineages in which these proteins are absent. Then we wonder if dicer and argonaut are being selectively maintained in Microsporidian genomes. We made a phylogeny with concatenated dicer and argonaut of 25 lineages. This phylogeny have inconsistencies with our species tree. This suggests that these genes are not being selectively maintained and evolve at different rates across lineages. Apparently, some strains lost argonaut while others of the same species kept both dicer and argonaut. We hypothesize that accumulation of TE in Microsporidian genomes is facilitated by vertical transmission. This mode of transmission reduces effective population size and the strength of natural selection, allowing the accumulation of mildly deleterious mutations. So we can see here that Microsporidia with vertical transmission have, on average, a higher proportion of these than species with horizontal transmission. Vertically transmitted species also have larger genome size than species exclusively horizontally transmitted. So in conclusion, the abundance varies greatly between species and it's not phylogenetically conserved. Vertical transmission is correlated to accumulation of TEs in Microsporidia, and genetic drift plays a strong role in the evolution of Microsporidian genomes. Thank you everyone for your attention, and I hope to see you in the poster session.